Bismillahirrahmanirrahim. Assalamu alaikum students. Today we are going to discuss the structure and germination of seed in Ephedra. The structure of seed shows in its longitudinal section that it consists of a dicotyledonous embryo in the center, and this embryo, as we have discussed in the development of embryo, is present at the tip of the elongated suspensor, and this embryo, it remains embedded in the endosperm. When the embryo is developing, the nucellus provides the food, so it is consumed, but it persists as a nucellar cap at the micropylar end of the seed. Seed is surrounded by the seed coat, which consists of two separate layers, which are produced from the two outer coverings or integuments or envelopes. And when the seed is mature, at the time of maturity, the subtending bracts of the megasporangiate strobilus, these become thick and fleshy, and these also form an additional covering layer around the seed, as in Ephedra foliata. This is the longitudinal section of the seed, in which in the central part embryo is present, which consists of two cotyledons. This is one cotyledon and this is second cotyledon, and this embryo is present at the terminal end of the suspensor, which is here, and nucellus is consumed while providing food to the developing embryo, but it persists as the nucellus cap to the micropylar end of the seed and ovule. And these are the two integuments, two envelopes. This is the inner and this is the outer integument, and these two envelopes, these give the two layers around the seed, and the bracts which are present around this ovule in the microsporangiate shoot strobilus, these also become thick and fleshy, and along with these two layers around the seeds, the third additional layer is formed by these bracts, which become also thick and fleshy. When the seeds are mature and these come out from the strobilus, then these germinate without undergoing any period of rest, means they do not go under dormancy or they become inactive for some period, but as they fall on ground without going any period of rest, they start to germinate if the environmental conditions are favorable for their growth. The germination of the seed is apigeal in Ephedra. Apigeal germination is that in which the germination of the seed takes place above the ground, as this diagram is showing the apigeal germination of the seed. The seed starts to germinate when it is present above the ground, and this germination is known as the apigeal germination, which is opposite to the hypogeal germination, in which the seed germinates underground. Now let's see the diagrams of germination of ephedra seed. This is the ephedra seed and it is testa of the seed. When seed starts to germinate, it ruptures at its one end to the micropylar end, and the part of the embryo which comes out, it is the hypocotyle. Hypocotyle is the region from which the radicle is produced. This elongates and this radicle is also developed and it matures and it produces the primary root. Here this diagram is showing the germination of the seed, longitudinal section of the germinating seed, hypocotyle region from which the radicle is produced, and radicle is the part from which the first root is produced, primary root is produced, and from this the uh, root cap will be also be produced along with the primary root. And these are the two cotyledons which are still inside the testa, inside the seed, and these are also coming out, releasing out from the seed as its germination goes on. Here the cotyledons are coming out, releasing out from the seed. These are the two cotyledons. In this diagram we can see that almost the seed is ruptured and the most part of the cotyledons is released from this testa. Hypocotyle is producing the primary root. Here we can also see in the next diagram also primary root is growing, it is developing, it is producing, enlarging in size. And these are the two cotyledons which are now, after releasing from the seed, are free. This is the remain of the testa which is still present at the apical parts of the cotyledons. And when these cotyledons become free, then in the axils of these cotyledons, cotyledonary leaves, here the epicotyle is present, and in this epicotyle region the plumule is developed, and from the plumule the shoot apex is produced from which the first foliage leaves are produced, and primary root develops from which the secondary roots are developed. And in this way, after germination, the sporophyte plant body is produced from the germination of the seed. These are the seeds of Ephedra sinica, in which the apigeal growth, apigeal germination of the seeds is going on. From the seed, hypocotyle region is coming out, and from this hypocotyle, the radicle will be developed from which the primary root will be produced. And then here are the cotyledon leaves which are inside the seed and testa, and after coming out of this hypocotyle region, then these cotyledon leaves will also come out. In this diagram we can see here that hypocotyle region, after producing the hypocotyle region will produce the radicle, and radicle will grow to form the primary root which will penetrate into the soil, and seed is still present at the upper region of the hypocotyle enclosing the cotyledons. Here is another seed from which the hypocotyle and radicle is coming out. And then in this diagram we can see that cotyledons are also released out from the seed. These are free from each other cotyledons, and from these cotyledons in the axils of this, from the plumary region, the first leaves of the foliage leaves of the sporophyte plant body are produced, and in this way the plant body is produced. In this way, from the germination of the seed, the sporophyte plant body of Ephedra plant is produced. In the next lecture, inshallah, we will discuss the life cycle of Ephedra plant.